Hello, Professor Sharon Schroeder here, March 29th. This is Lab 5, the bacterial morphology staining technique results video. Um, so again, I have my rubric here of the points we're going to be that you want us to go over for the results video, as well as I'm going to show you some of the results that I have already put into the hands-on lab data table. So I'm going to begin by, let me bring up this. So for the exercise one, the direct and negative stains, this is where we were first introduced to heat fixing, which is a process to get the bacteria or whatever you're going to be using to be fixated onto the slide so you can view it. That was this whole exercise one, was pretty straightforward and self-explanatory. Let me see here. Let me bring up my data table to show some photos from exercise one. So in this one, we use the Congo red and the what is it? Something violet. So right here you can see the this was from the purple stain, and then this is my, let me bring up the other one, my slide from the red stain. So on the one that I used, the Congo red, I don't know if I left it on too long or if I put too much, but it looked like it was almost like a watercolor paint. I don't know if that's how it's supposed to look usually, but that's how it came out for me. Let me see, and I did follow all the steps pretty completely, or maybe I just didn't allow it enough time to sit. So in this whole lab, what we're learning is the cell morphology of the different bacteria. So the three main bacteria shapes are the coccus, the bacillus, and the spirillum. So it's it was I think that was important to cover, because sometimes when you look at the slides you just think, oh, there's this little squiggly liner, but knowing the different types of shapes and arrangements, I mean, that this is like, of course scientists know this, but as a normal, like, just student, you wouldn't know what something is. But knowing now that we've learned about this, the arrangement of the bacteria, the size, the formation, you can kind of, once you get something under a microscope, figure out what certain bacterias are. So I thought that that was pretty, it was pretty interesting to learn about. Let me see. So that was for that exercise one. So let me go to exercise two really quickly. So for that, so really quickly to just recap. So on the first one, we this is where we did let me make sure, heat fixed the specimen, but let me make sure, I forgot what we what we, I think it was like a sample from our teeth or something. I've just been going through it, but yes, so it was a sample from the teeth and the gums, and then we stained them with the Congo red and the crystal violet, and then after that we let them sit, and then viewed them under the microscope to see the morphology of these specimens. So that was for exercise 1. Now exercise 2, let me bring this up. Exercise 2 was the differential stains. Let me bring this down. So for the differential stains, this one we used the already made cultures that we had from our second lab that we did, and we got the cultures, got an inoculation loop, grabbed a sample from the culture bottles, smeared them on a clean slide, heat fixed them, and then let me see here. And then from there, we did our gram staining process. So it was a pretty lengthy process. It wasn't like you could just drop them on there. And so like for the first couple of stains for the let's say for the crystal violet one, you had to stain the specimen and wait 60 seconds and let it sit, and then you move on to the next one and the next one and the next one. So I feel like it was pretty lengthy in time, the experiment. Let me see here. And then for these ones, after that, we, of course, viewed them under the microscope. But let me bring up my data table to show you just. So this is from, let me make sure I have, this is from the E. coli. And of course, you have all access to all this information, so I'm just sharing this with you. So the gram staining process was pretty interesting to learn about in and of itself, learning that the difference between gram positive and gram negative. Gram positive cells, you can tell because they are thick celled walls. The gram negative had thinner cells. They don't have the peptidoglycan layer. So those are more resistant to antibiotics. The antibiotics can't go through the thin cell wall, so bacteria such as E. coli and other bacterias that can't be, they're gram negative bacteria, they can't really be penetrated. Those are more susceptible and harder to treat with antibiotics. So let me just make sure I covered everything here. So yeah, so really quickly, just to backtrack, for each one of the cultures, we had to do one of the slides the same way, same process. So again, this was a very lengthy experiment. Let me make sure. So, explain whether the experiment was successful, whether expected results. So under the microscope, like I had explained in my earlier results video, I feel like I was still in a learning curve on how to use the microscope, so I don't feel like I got the best views of the cultures that I probably could have. So of course I'm gonna work on that. But I did see, especially on the yeast one, you could really see the formation and the arrangement of the specific bacteria. So it was pretty interesting on that. Explain how the experiment results apply to real life. So for gram staining is helpful. It's like helpful because it facilitates which antibiotics needs to be used when there has been and there is an infection present and you have identified what bacteria. So it helps to identify that bacteria so you know what right antibiotics to give. Like, let's say if you're a doctor or patient relationship type thing. um what else do I want to say? So I think that is one positive way that they are used in real life. Describe any experimental errors that occurred. The only thing I would say is that maybe I didn't leave the stain on for long enough on some of the specimens, so that maybe next time if I have to do this, I will have a designated stop clock. But it's kind of hard to do all that when you're trying to make sure you're not contaminating anything, and it's just a lot to make sure that everything is the way it's supposed to be going and keeping time also. So I think I pretty much went over everything, so that is it. Thank you.